Hello everyone, we are now going to go over the peripheral blood flow cytometry from a 63-year-old woman with neutropenia and lymphocytosis. So let's start with CD45 versus site scatter. When you see CD45 versus site scatter, you have a large number of cells in the lymphocyte gate, I think 64% of cells with bright CD45 and low site scatter, and monocytes account for 1% of all cells, and they have bright CD45 and slightly higher site scatter than the lymphocytes. Granulocytes express low or dimmer CD45 than lymphocytes and monocytes, and they have very high site scatter, and there is essentially nothing in the blast gate. And the lymphocyte gate is composed of both T cells and B cells. The B cells are CD19 positive. If we track them, they have small spray of CD5 like you usually see in the peripheral blood. They have partial 23 expression, FMC7 positive, negative for 200, all normal, and they have a nice polyclonal kappa lambda ratio. So our B cells look fine. And there are CD5 positive cells here that are the T cells. And normally when you split the B cells and T cells, especially in 19 versus 5 plot, majority of the cells would be positive or 5. Because those are T cells, and then the 19 positive cells will be negative for CD5. That's how normal peripheral blood lymphocytes, appear in this scattergram. But here we see a large population that is negative for both 19 and 5. And these tubes, these markers in this tube does not further characterize it. We have to go to the T cell tube to see what they are. Now, when we go to the T cell tube, we again see a small population of CD3 positive T cells, and the larger subset is positive for 7. There is a small subset of CD3 positive cells that are negative for 7, but that's something that we usually see, not a problem. But there is a large population that is CD7 positive and negative for CD3. So those could be NK cells. We can keep looking, and those are negative for CD5. So this is what we saw in the previous tube. These are positive for 2. Again, NK cells can be positive for 2, and these are positive for 4 and 8. And negative for 4 and 8. 4 and 8 positive T cells are seen in the background, and these are all negative for CD10. And in 57 and CD3, we see that these are really bright for CD57. So CD57 is one of the NK cell associated marker, but 57 is more often expressed by LGLs than NK cells, but NK cells can also express 57. So the additional tube containing 56, 57, and 16 is now, we are seeing that. And if we see that our population of interest does not express 56, it expresses 57 and 16. So it has the NK cell associated antigens 16 and 57. There is no surface CD3. It is negative for CD5. It's only positive for 2, 7, 57, and 16. So normally you are tempted to think that these are NK cells. So it could be NK cells can have this combination, but we have to remember that the literature says 100% of LGLs or T cell LGLs are positive for surface CD3. But the ultimate thing that is going to distinguish between the surface, between the LGLs and NK cells is not the surface CD3, but the T cell receptor gene rearrangement. So we have to send these cases for T cell receptor gene rearrangement. So I have to admit that when we initially got the flow for this case, we thought, oh wow, it's negative for CD3, it is positive for 2 and 7, it has 16 and 57, so it's probably NK cells. And we the patient has been having this persistently for years, and clinically they called it chronic lymphoproliferative disorders of NK cells, and the patient actually satisfied the criteria for it. But when we subjected this to PCR, we found that T cell gene rearrangement was positive. So then we reverted this to be just LGL proliferations and not NK cell proliferation. So I want to emphasize that when you see something like this, although it looks like NK cells, you just have to do your PCR to differentiate between the two. Some labs perform flow cytometry for killer cell immunoglobulin-like receptors for NK cells, because just like you see kappa lambda light chains on your B cells, or alpha, beta, and gamma delta on your T cells, NK cells, although they do not have T cell receptors, they do have KAR receptors, and they can, a restriction of these can indicate that these are neoplastic. So some labs perform KAR flow cytometry, although we did not do that here. So the take-home point in this one is when you see something like this, when you see both an LGL proliferation or an NK cell proliferation, it's always a good idea to do a PCR to make sure that this is actually the cell type that you are calling it, and also it is clonal. The other thing that you want to make sure is do not call them as leukemia in your diagnosis. Whenever you see increased LGL proliferations or NK cell proliferations, just indicate the percentage of cells that you see and what phenotype you see, because there are a lot of clinical factors that need to be considered to call these as leukemias. Thank you.